வணக்கம். நம்ம ப்ரீவியஸ் வீடியோவில் பயோசிந்தட்டிக் பாத்வேவை பற்றி பார்த்தோம். நம்ம இன்னைக்கு பார்க்க போகிற வீடியோவில் கோலோபிளாஸ்ட்டை பற்றி பார்க்கலாம். வீடியோக்குள்ளே போகிறதுக்கு முன்னாடி
This video is sponsored by BuyMote shopping app. BuyMote e-shopping application is one of the online shopping app. நீங்கள் உங்கள் வீட்டுக்கு தேவையான டெய்லி நீட்ஸை இந்த BuyMote e-shopping ஆப்பில் டிஸ்கவுண்ட்டோட பர்ச்சேஸ் பண்ணிக்கலாம். அது மட்டும் இல்லாமல் BuyMote வெல்கம் கிட்டுன்னு ஒரு காம்போ ஆஃபர் போயிட்டு இருக்குது. 999க்கு 25 ப்ராடக்ட்ஸ் கொடுக்குறாங்க. எனக்கு அது வர்த்தாக தோணுது. உங்களுக்கு வர்த்தாக தோணுச்சுன்னா ட்ரை பண்ணி பாருங்கள். இதில் பாப்புலரான பிராண்ட்ஸும் இருக்குது. நமக்கு தேவையான பிராண்டுக்குள்ளே போயிட்டு நம்மளுக்கு தேவையான ப்ராடக்டை டிஸ்கவுண்ட்டோட பர்ச்சேஸ் பண்ணிக்கலாம். இந்த ஆப் ப்ளே ஸ்டோர் அண்ட் ஆப் ஸ்டோரில் அவைலபிளாக இருக்குது. ஆப்போட லிங்க் கீழே டிஸ்கிரிப்ஷனில் இருக்குது. ட்ரை பண்ணி பாருங்கள். வாங்க இப்போ வீடியோக்குள்ளே போகலாம்.
கோலோபிளாஸ்ட்னா என்னென்னா பிளாஸ்டிட்ஸ் ஆர் எண்டோபிளாஸ்மிக் ஆர்கனல்ஸ் ஆஃப் photosynthetic proteins and plants. Possess pigments such as chlorophylls and cathodes are the sites of synthesis and storage of food reversers. The most important type of plasmid in the chloroplast. Chloroplasts contain a chlorophyll and use a light energy to convert carbon dioxide and water to carbohydrates and oxygen, sites of photosynthesis. Chloroplasts variable in size and shapes, oval with dimension of 2 to 4 by 5 to 10 by some algae forces one huge chloroplast. Like mitochondria, chloroplasts are encomposed by two membranes. A matrix called the stroma is enclosed by the inner membrane. It contains DNA, ribosomes, lipid droplets, starch granules. A complex internal membrane system whose most prominent components are flattened membrane delimited snacks called thylakoids. Cultures of two or more thylakoids are disappeared within the stroma of the most algae chloroplasts. In some prokaryotic hosts, several distinct thylakoids are stacked on each other like coins to form grana. Photosynthetic reactions are separated structurally in chloroplasts. The trapping of light energy to generate ATP, NADPH and oxygen is refers to the light reactions. These reactions are located in the thylakoid membrane where chlorophyll and electron transport components are also found. ATP and NADP are found by the light reactions are used to form carbohydrates from carbon dioxide and water in the dark reaction. The dark reaction takes place in stroma. External cell coverings. Eukaryotic microorganisms differ genetically from prokaryotics in the supporting and protective structures. They are external to the plasma membrane. Many eukaryotics lack an external cell wall, example amoeba. Eukaryotic cell membranes contain sterols such as cholesterol. Many eukaryotics do have a rigid external cell wall. The cell wall of photosynthetic proteins have a layered appearance and contain large quantities of polysaccharides such as cellulose and pectin. Inorganic substance, silica in dimetals or calcium carbonate may be present. Fungal cell walls normally are rigid. The external composition varies. Cellulose, chitin or glycan are present. Rigid materials in the eukaryotic cell walls are chemicals simpler than bacteria peptidoglycan. Many proteins have supporting mechanism called the pencil. The relative rigid layer of components just beneath the plasma membrane. The penicillin may have simple in structure. Illuna has a series of overlapping strips with rigid and the edge of each strip fitting into the groove of adjacent one. Pencils of seeded protozoa are exceptionally complex with two membranes and variety of associated structures. Although are not strong as rigid cell walls, they give the precursors or characters shapes. Cilia and flagella prominent organelles associated with the motility in eukaryotics. Both whip like a bee to move the microorganism along the differ from one another in cilia 5 to 10 in length, whereas flagella are 100 to 200 long. By the patterns of movement usually distinctive. Flagella move in undulating fashions and generate planar or helical waves originating at the edge of the base or the tip. The wave moves from the base to the tip, the cell pushed along. A bee traveling from the tip toward the base pulls the cell through the walls, femur filaments. Sometimes the flagella has later hair called femur filaments. Thicker, swifter as are called mastocymias. These filaments are change of flagella actions so that wave moving down the filament toward the tip pulls the cell along the instead of pushing it. Such flagella often is called tisnal flagella, whereas stacked flagella is referred as whiplash flagellum. Cilia. Cilia have a bead within the two distinct phases. In effective stroke, the cilium strokes through the surrounding the fluid, thereby propelling the organisms. The cilium next bends along in the length while it pulled forward during the recovery of stroke in preparation in other reflective stroke. A ciliary microorganism actually coordinates the beats so that some of the cilia are in recovery phase while others are carrying out their effective stroke. This coordination allows the organism to move swiftly through the water. Despite the difference, cilia and flagella are very similar in ultrastructure. Now, Bamarine bounded the cylinders about 0.2 diameter, located in the matrix along the axis which one 9 pairs microtube doublets arranged in circle around two central tubes. This is called the 9 plus 2 pattern of microtubes. Each doublet has pairs of arms protecting from the subtube, a complete microtube towards the neighboring duct. A radial spoke extends from a subtile toward the external pair of microtubes within the central sheet. These microtubes similar those who found in the cytoplasm. A basal body lies in the cytoplasm at the basal edge of cilium or flagella. It is a short cylinder with 9 microtube triplets around the pipillary 9 to 0 pattern and is separated from the rest of the organelle by the basal plate. The basal body derives the construction of these organelles. Cilia and flagellum appear to the grow through the addition of performed microtube subunits and their tips. Okay, thank you.